Yeah, so for your unknowns, you can do the lab work up through next Friday, which I think is the 18th. And so that, you should plan on having all lab work done by 5 on Friday the 18th. Because then there are not going to be many people around during Thanksgiving. And Charles won't be here and I won't be here and doors will be locked most of the time. But then the reports are going to be due the Monday after Thanksgiving. And those you will turn in on Canvas. Actually, so you don't have to print them out and bring them to me. There'll be a place to upload it on Canvas. Which is good because that way you don't have to print color, and I tend to grade written work faster if it's on Canvas. So I don't have to carry the papers with me and stuff. So that's for your unknowns part. Did that answer what your questions were? And then for your parasite presentations, those are going to happen Tuesday during lab. And I actually have to figure out like what room are those happening in, because Dr. DeFord has a class in here during that time. It's possible that they might be in that little room across the hall over there, because I think we can fit in there and that would work. Okay, so for those are going to be a PowerPoint. If you love Prezi and you want to do a Prezi, that's okay also. I feel like that was a fad. You know what I mean? Prezi. Anyway, kind of like PowerPoint, but different. And so those, you don't upload the presentation. You just come in and you give the presentation. But the study sheet thing that John asked about, that is a little bit different. So your study sheet would need to be uploaded to Canvas. And so I may not have that upload ready yet, but that will be a file upload. And then what I do is I take those files and I put them in a folder or like a module, and that already exists. And then you all can review over those to study for the final. So on the final, there is a big matching of parasites, and it's like, this is the name, the scientific name, and then there's a description. So these study sheets, you upload that to Canvas, and that's 5 points. And then the presentation ends up being 95 points. And I think you all have the rubric, and the rubric's also on Canvas, so that's what I use to grade. And I just sit there. Charles may also grade. Probably going to have him fill out grade sheets, and then I'll look at it, and if he's reasonable, maybe we'll average, but that's what I use to grade, is that rubric with the points. So.
What happens in like
I like that question also, because this happens sometimes. The question is, if you didn't hear it on the video, what if you identify your unknowns and it really didn't take that many tests to do it? It seemed like not maybe enough. So what I looked for on unknowns is kind of this. Did you do all the usual tests for that category? So for example, if you think that your unknown is a Streptococcus, and then you would want to have included every test that you would do on Streptococcus. So the BIOL esculin slant, the catalase, the CAMP multi-test. Maybe that's, I feel like I'm missing one. But likewise, if it was Staphylococcus, you would need to do all the Staphylococcus tests, even if you think that the results that you've got off of these two tell you what it is, do the other ones also. Or on the gram negative, if you think you have a gram negative, do all the gram negative tests, even if you think you don't need that one. Just to be sure. But yeah, I know I said don't waste, but you can do all the tests in a certain group Because that's not really wasteful. That's just confirming what you might already. So those, your charts, those table things where you all fill out this strain and this result, that's gonna have what tests you need to do. And then kind of your notes from class also, if you think you might be missing them. Really good questions, because people need to know that every year, and I forget to say it sometimes in the beginning, like, you should do all the tests, even if, like, for example, even if you've got, like Salmonella, Typhimurium, and you would expect that the McConkey auger is gonna be negative, you should still do it. That'll just confirm, it actually becomes more evidence that, yeah, that's what you've got. Good questions, good questions.
Is it okay if we use like a chart? to show?
Yeah, I have had people use charts.
I was just wondering because I've had like this. Pictures with it.
Yeah, I want some that's like writing. Like some writing in sentences, but I've had people do effective use of charts like that to just show everything. And if you've got, the place where a chart will not give enough info is for things about how the test works. For example, if you had in that chart like a McConkey auger, probably you need some written description later of McConkey auger test for lactose fermentation, and if it, methyl red is the indicator and so on. Lactose is fermented to acid. So for those, you probably need some writing. But I can imagine a lot of other things you could put into a chart and have that included. So as long as you've got, what did you do? Why did you do it? What was the incubation time? What does it let you infer? I feel like that could fit into the chart. How the test works, maybe might have to be outside the chart. But any questions like that you have about how to write your report, or if the format is right? I don't give a lot of format details because I feel like you get so many specific format requirements for specific writing tasks, and this one is a little flexible as long as you got the info. Yeah, good. I have discussed, I don't know when this started with Teams, but it now makes a transcript of everything that you say. It tries to. And this morning in AP2 lab, it was reproductive anatomy. And this is so funny. I'm going to print it out and read it to myself on days like a needle laugh. It's ridiculous. So like oocyte, it can be OS site opposite suicide, like how it's so silly. And then just the stuff I was talking about is also funny in a way. So those of you, if you're watching this later, there is a transcript of what I say, but I feel like you should just watch the video because the transcript is going to come out weird. So goal for today is to learn basic details about the eukaryotic parasites. Yeah, I am recording. And we've got really 3 big types that infect people. And you could just circle them if you've got these, or make a list if you don't have the picture. So we got protozoans, fungi. Algae so far do not infect people. They do not get inside us and cause an algal infection of a person. And then we have the helminths, which are parasitic worms. Some of you, for your unknowns, or not your unknowns, your parasite presentations, your parasite is actually a bacterium that is carried by an animal. And hopefully, if you've got that situation, you already know it. But there's going to be a part on your parasite presentation where it says, what type of parasite is this? And it's not ever an insect or a tick or mosquito. That's not the parasite. They're carrying one of these other things. So bacteria, if it's carried by an animal, put into the person, it's not included in this group, but some of you have one of those assigned for your parasite presentation. So our info today is going to be about protozoans and fungi and helminths. And all of these parasites are eukaryotic. Everything we've learned about thus far in class has been a bacterium, which is a prokaryote, or a virus, which is neither thing, but we're now kind of turning back to eukaryotes. And this is just to remind you all the stuff that a eukaryotic cell has. And this particular eukaryotic cell has a flagellum, so it's not human. This would be some kind of protozoan. So this group of parasites has all of the same cellular equipment that we do as their host. And that has a lot of implications for how we treat these infections, because these parasites are very much like us. So at a cellular level, eukaryotic cells, eukaryotic parasites, quite similar to our host cells. We share a lot of machinery. So you can think about eukaryotic cell stuff in a picture form or in a word form. And this is a word flowchart form of, okay, what do eukaryotic cells have as a huge group? So this is not what human cells have. This is what eukaryotes as a domain of life can have amongst themselves. So they can have flagella. They can have glycocalyx sometimes. They can have cell walls sometimes. They always have a cytoplasmic membrane. They've got cytoplasm. They have a nucleus. So this is the traditional marker of what is a eukaryote. So it has a nucleus and then it has organelles in the cytoplasm. It has ribosomes and then it has cytoskeleton. So kind of in this list of stuff here, this level, which one of those things can prokaryotes also have? Cytoplasm. So this would be shared by both. So anything I put a check mark, both domains of life would have it. What else? Ribosomes. Not organelles as much. Like that's more of a eukaryotic thing. Cell wall. Flagella. Cilia are eukaryotic. So they look similar to a fimbriae.
Oh, that's.
They look the same, but they're actually really different. And like we haven't learned enough about what fimbriae are and what cilia are to know that. They do look the same, they're actually different. The other ones that both have are membranes. And they actually both have cytoskeleton, but we haven't learned that much about that in this class. So there's some parts that can be shared that both domains of life have. Some of them, just because a eukaryote can have it, doesn't mean it's the same version. So flagella exist in very different versions in bacteria. Cell walls exist in very different versions. The membranes are pretty much similar. A membrane is a membrane, no matter where you find it. The cytoplasm is similar, both. Ribosomes have slightly different versions. And the reason I make a big deal about this is because when we make drugs, we're gonna make drug treatments to treat infections. Those drug treatments should target structures that are unique to the pathogen. And those structures should not be shared by us, because if we have that same target, then the drug is going to harm us. For the end of next week, we start talking about drug treatments. So we have to target what is different to develop good drug treatments so that we treat the infection without harming the host, which is a person. So that's why knowing what the parts are and what's different is very important. The flagella in eukaryotes and the flagella in prokaryotes both have a similar purpose to move the cell, but they are actually really, really different. They're made out of different pieces and they actually have different motions. So the prokaryotic motion, so here's bacterium. Prokaryotic motion of a flagellum is to spin. And here's a bigger cell, eukaryotic, with the eukaryotic flagellum. And the eukaryotic motion is to wave. So kind of do a back and forth wave like a fishtail. And they're both flagella, but they're actually vastly different from each other. This would be, well, move on. Both have flagella, can have flagella, but they're really different. Likewise, cell walls can exist in both fungus and bacteria. And they're serving the same purpose, but they're vastly different in what they're made out of. So this picture is showing fungus. So this is fungal, and this is fungal. And fungal cell walls have as their major building block is something called chitin. So for a fungus, chitin is the major building block. What is the major building block for bacterial cell wall? It's basically made of. Peptidoglycan. Yeah. So they both have walls, but the walls are built and constructed differently out of different chemicals. Protozoans and helminths don't have cell walls. Plants do, but they don't infect us. So fungus, bacteria, they both have cell walls, but they're made out of different chemicals. All life forms, all cells, so all life is cellular, all life forms are cells, made out of cells, and they all have ribosomes to build proteins. So the ribosomes are going to be essential in any of these life forms that we look at, you have to have ribosomes. They are essential to build proteins. But the prokaryotic ones are differently shaped. So they're slightly different. They're a little bit smaller. They're a little bit shaped differently. And this side is prokaryotic. This side is eukaryotic. So you kind of draw a line down between here. Oh, it says it down here. Like they're basically the same. They got similar shapes, but not exactly. So the key fact about this that relates to treating infections is that we can make drugs target the slight differences. In the prokaryotic ribosomes. So when we start learning about types of antimicrobial drugs, there's really a lot of them that target ribosomes. Because prokaryotic ribosomes are enough different from ours that we can make a drug to stop theirs, leave ours alone. But eukaryotic ribosomes are similar enough to each other that we can't make a ribosome drug that would kill a worm, but not us. They're too similar. So we both have ribosomes. Eukaryotic ones are real similar to each other. So that's kind of the first section. So what we're going to do next is look at each big group. So we've got fungus, protozoans, and then worms, and kind of just say some real basic things about how they act. 3 of you have had Bio 2, right? You 3 gentlemen, did you have Bio 2? Okay. I asked Con, he didn't remember you, so maybe you didn't have it. So you 2 have had Bio 2, and like probably know all this, but it's a good review. Now think about it from a medical sense. So we're first gonna look at fungus. The good news about fungus is that there are relatively few in the whole world of fungi that can infect people, which is good because there are a lot of fungi in the world. We have relatively few that are able to infect humans. And because of that fact, there are actually way more fungi than we realize or think about because they don't usually affect our life. And when you have learned about fungus before somewhere, you'll recall that there is a part of the fungus that exists as hyphae, or these thread-like structures that grow into its food source. And then there are these reproductive structures that they make called fruiting bodies, and those have spores that they release. Raise your hand this week if you needed to use bread that had mold on it. Because you're running out. No? Go eat the calf, they will give you moldy bread. So most important to think about for fungi that do infect people is that the spore drifts away in the air, but this is not like an endospore. They are not endospore level of tough. These are actually quite wimpy. And they're actually pretty easy to destroy, and they float through the air to spread fungi to new locations. So when we think about preventing people from getting fungal infections, a lot of times we're trying to stop them from breathing the spores. So most of the fungi that we are familiar with, you get a spore, it lands on the item, like strawberry or bread, and then the spore germinates, and the fungus starts building hyphae and it spreads through the surface and they grow down. What they're calling submerged hyphae. So they would be growing down into the food source. And when the fungus has grown to a certain level and it's absorbed enough nutrients, then it produces these treating bodies that then release spores, and the spores go and land on a new place, and this cycle continues. That's familiar. Now, what's kind of gross to think about is that if fungi are growing on the surface of the body, they are sending hyphae into the food source, which is your skin or your nails or your hair. That's for fungi that are growing outside the body. We're going to see some examples of fungi that get more deep to the body, deep into the body. But if the fungus is growing on the surface of a person's body, it's basically the hyphae that are doing tissue damage and growing into skin or hair or fingernails, maybe. So here's some examples. And some of the diseases that people get that are fungal, their name doesn't say fungus. It has some other name that makes us think that it's not a fungus, but it actually is. This is hair fungus. Do you know fungus could grow into your hair? It can. It doesn't often happen in this country, fortunately, but grow into your hair and absorb nutrients from your hair, and it can make your hair break off. Not a huge medical concern, but it's just kind of gross. What is this? If you saw that in somebody's foot, you'd say, dude, you have athlete's foot? Yeah, athlete's foot. The disease that people call jock itch is also fungal. Tends to look like that in a different location. This condition is kind of a very advanced version of what we call ringworm, which is not a worm, it's a fungus. So any disease that has kind of like a circular patchy, not any disease, the several diseases that are circular patchy appearance on someone, the common name for it is ringworm, but it's actually fungal. So this is ringworm. And this one is different from these others. I'll write the word of it over here. The one in bottom left, its common name is thrush, like the bird. There's a bird called a thrush. Thrush can also be a disease. And what thrush is an oral yeast infection. Where the yeast is overgrowing on the surface of the person's tongue. So in your nursing career, you'll tend to see thrush on patients that are immunocompromised, and it's their own normal yeast flora overgrowing. And you tend to see it in very young babies because their immune system's not fully active yet. But that condition is called thrush. So all of these are surface infections where we've got fungi on basically the outside of the body, but it's not infecting the deeper tissues. Most fungal infections in people are superficial, surface level, and they're not reaching deeper tissues. And these are unsightly, and they make people feel bad, and they make people look bad, but they are not usually major health threats. Fortunately, most fungal infections are superficial. There are some fungal infections that are not superficial and that cause systemic or deep infections. And these are caused by what we refer to as true fungal pathogens. These are less common, but they are more damaging to people when they contract them. And those of you that are presenting on fungal parasites, you have these because these are the ones that really harm people's health more than the superficial ones. But you'll see the superficial ones way more often when you're doing nursing. So to be a true fungal pathogen, the fungus have to have some particular characteristics, which are they must be able to survive body temperature, which is fairly warmer than the average outside environment. That's why not that many fungi can infect inside the body. And they also must be able to reproduce without their hyphal development stages. And not that many fungi can do that. So if you're floating around in body fluids, you need to be less dependent on hyphae for your life cycle. And these characteristics are basically referred to as thermal dimorphism. If you're going to infect deep tissues of the body, you have to be able to be a little bit more flexible and not just grow as hyphae, but be able to survive fairly hot temperatures in a person and basically exist in like a yeast form. So some fungi don't have to go through really a hyphal stage, but they exist more as just individual cells and they bud off. And that's yeast form behavior. Not many fungi can do that. So on Tuesday, you'll learn about a couple of examples of true fungal pathogens that can do this, and they can infect people and cause them some more serious damage. Here's the yeast representatives. Sorry, yeast. Who has a fungal disease for your parasite? Probably one more. Might be one of those ladies. Who's got worms? And then protozoans? Yeah. And then a couple of bacteria carried by ticks. Yeah. Yeah. So one of them has worms and one a protozoan. I'm not really going to say anything about this. Like, that's pretty understandable. If you've got a surface fungal infection and it stays on the surface, it's annoying, but not that bad. If it gets into deep tissues, it's going to be harder to get rid of.
So does helmets have to do with that word?
What's the first word?
Helmets.
Helminths. Yeah, helminths are the technical name for the worms.
Because I was confused when I was with them,
Mhm.
because I couldn't tell if mine was a protozoa.
Or a helminth.
But
You figured it out later?
yeah.
Yeah.
I finally figured
Yeah. Yeah, helminth. That's a hard word for me to say. Get that lymph word in there. Something I want to use as our last slide on the fungi is to point out some root words that when you're reading the name of a pathogen, how you know it's a fungus. So if they have the word in it, it's not tricho. If it has the prefix or the suffix phyt, P-H-Y-T, that is a fungus. And that shows up in a lot of different fungal names like trichophyton, epidermophyton, different diseases. The other term that tends to indicate a fungus is this, says spore. Something sporum, sporobothrium or something like that. That's an indicator that it's a fungal disease also. And then the other words can tell you trichophyton, that means it can infect your hair. It's a hair fungus. Or a skin fungus. Microsporum, it has really little spores. Epidermophyton, that's a fungus of the skin. Do you all remember, you would have been little kids, there used to be a commercial for a nail fungus treatment. And it was this little cartoon, it was called Digger the Dermatophyte. He would like get in, and it would have this cartoon of like someone's toenail lifting up like a trunk of a car, and it would jump in there. It was so gross. You remember this? It may have been too long ago. It was, to me, the most disgusting medical commercial I've ever seen. It is a cartoon. Digger the Dermatophyte, and they were trying to sell medicine for toenail fungus, which is hard to get rid of. You gotta basically wait for your nail to grow out before you completely get rid of it. Okay, so our next group of eukaryotic parasites are the protozoans. And they are really diverse as a group. So they, as a group, they're eukaryotic, they're single-celled, and they can move themselves from place to place. They don't have a cell wall. So that's what kind of Sir Han and Professor Rudd would be like clutching their pearls right now. That's what protozoans are like. And they have different ways of moving themselves from place to place, which puts them in categories. So if they crawl, they're an amoeba or amoeboid. If they swim by cilia flapping, they're ciliated. If they move by a flagellum, that's a certain category. And then these other ones just kind of wiggle from place to place. They're kind of moving by some other mechanism. So they have different categories, but all of them have in common that they're going to live in a watery environment. So they are small eukaryotes living in watery environments, and they can move in some fashion. If you had Gen Bio with Han, he is more algae and fungi focused than protozoans, I'm pretty sure. Oh, you're in plant? You were in plant? Yeah. Moses came to algae lecture last week. And I told him, it's like, this is the most you'll learn about algae ever in your life, probably, unless you become an algae specialist. So protozoans, I like organisms that move themselves. Protozoans are a real diverse class, but there are some of them in their life cycle, they kind of go through two formats called cyst versus trophozoite. So this is showing for some general protozoan that it can alternate between a version called a cyst. And so the cyst is a little bit tougher. It's round. So it's round, it's a single cell, and it's got a tougher covering on it. And this conceptually is kind of like an endospore, but way less tough. So it's tougher than the trophozoite form, and it can survive longer outside of watery environment. But the cyst, when it reaches a good environment, it will start dividing, and it will form the version of the organism called trophozoite. So the trophozoite can move, eat, kind of do normal things that you would expect a living cell to do. So it's kind of the more animal acting form, and the cyst is basically sit, wait, survive. Most people, when they catch a protozoan infection, they are actually getting the cysts. So they swallow the cyst, and then the cyst moves till it's in a good spot, which is the digestive tract. So cysts can make it through stomach acid and still survive. And then when they get down to the digestive tract, that's good times. It's warm, it's wet, there's food. Time to party, it's time to be a trophozoite. And in the small intestine, you will have trophozoites swimming around and stealing your food and maybe interfering with your digestive function. But then when they get carried to large intestine, and that's less good times and it's kind of shitty. They turn back into cysts and you poop out the cysts and then the cycle continues. Most protozoan infections that people get, it's from waterborne infections in some way. So drinking contaminated water or swallowing contaminated water is one of the major ways. And the second major route of getting protozoans is insect vector delivery. So some protozoan infections, you get it basically from drinking dirty water, ingesting cysts from fecal contamination, or an insect transmitted a protozoan into your body. Those are the three major ways. Any of you all assigned insect vector protozoan? Maybe not. I think not this year. There's one from a cat. There is at least one from drinking the water. There's two, and there's one from water up your nose. Should be. For y'all's presentations. This is kind of showing what animals can pass you protozoans, what animals can be vectors. It's really a lot depending on which disease we're talking about. So animals are really frequently involved in passing people helminths or protozoans. And probably one of the worst protozoans that you can get in modern times is the one that causes malaria. We don't have people in the US getting malaria very often, so I did not assign it as a parasite presentation, but you need to know this basic stuff about what malaria is. So when people get malaria, they're getting this protozoan parasite from a mosquito. So the mosquito bites them. It has the parasite mouth parts and it transmits to the person. And then this Plasmodium falciparum parasite infects the blood. Yeah, affects the blood. And malaria is the most deadly childhood disease that exists on Earth right now. It's really hard to stop it. It's really hard to stop it. You had a single wish to improve health of kids all over the world. Stopping malaria would be a good candidate. And then the second one would be having enough food for kids. They're equally serious issues. So we'll spend the rest of our time today on helminths. Helminths. Wanted to use this slide to show a little bit about what are the major body plan varieties of helminths. There are basically three, and one of them's not shown on here. So I'm going to draw one on, trying to indicate body plan number 3. So the first type of body plan is called roundworm. And if you go out and dig worms to go fishing, that's a roundworm body plan. So it's kind of tapered at both ends, and it has like head to anus construction. And if you slice through it, it would be basically a round cross section. That's roundworm. There are also flatworms, or flukes, that are, if they're lying on the surface, they're flattened. So if you did a cross section of them, it would look like this. They're not round in cross section, they're flat. That's fluke or flatworm body plan. And then we have the segmented or tapeworms body plan. These two act more like a normal animal. All worms are animals. All these are animals. The tapeworms, if you just have one of the sections, each section has eggs. And so these can break apart and you can ingest just a section and it will still infect you. I find that disgusting. So for me, worms and blood-sucking insects are the scary type of infections to get. I don't really have to write anything about that, but. So those are 3 types of worms. Not every type of worm infects people. Just like not every type of protozoan or fungus infects people. This is the body plan of a roundworm. So we could just write here. This is basically roundworm structure. And on this worm, this type of worm, males and females have different lengths and slightly different anatomy. When I first started teaching this class back in Arkansas, they wanted students to be able to identify worms off of a slide and know, like identify cysts of parasites and stuff like that. And I thought, whoa, that's intense. So when I had freedom to make the class the way I wanted it, I cut that stuff. But I was thinking this year, I really ought to at least put the slides back for people to look at because it's cool. Don't identify them. This chart is just informational and not something to study, but it's just showing, these are some major names of worms that infect people. What's the disease called? What organisms does it pass through and how do you catch it? So I want to focus more on how do you catch these? Most of the time, we catch worms by ingesting worm eggs. Ingesting worm eggs because the eggs, again, are tough enough to make it through the stomach, and then they tend to hatch in your intestines and you have intestinal worms. So that's one major way to catch a worm infection is to ingest worm eggs. And usually the ingestion of worm eggs is by fecal oral mode of transmission. That's one way to catch a worm infection. The second way, this is like the third way, it's very minor. Second way that's common to catch a worm infection is to ingest them in uncooked meat. Or let's not say uncooked, let's say improperly cooked. And this can include any type of meat, fish, seafood, and also snails, which I feel like don't go into those other categories. So that can be a way that you can contract a worm infection. And I know one of your worms that you're assigned is contracted through the improperly cooked meat. The third way, which is terrible, but fortunately rare, is that worms can burrow directly through the skin. That version is more rare. There are not that many worm infections that you can get that way. But I remember when I was a kid and I heard of that concept, like if you go barefoot in the grass, there can be worms that crawl up through your foot. And I wouldn't go barefoot in the grass for many years after that. So I realized at some point, I have never heard of this happening to a real person that I know. Maybe it's gonna be okay. Those are three major ways to catch worms. And I think I know that's so fast, and it's just barely any info about any of these, but that's enough that when you hear people's parasite presentation, you can understand what they're talking about, and they'll help you understand your presentation a little bit better too. So for Monday, we will start a new slide set that I need to develop over the weekend about how to kill microbes. That's what's coming up next. Everything from now to the end of the semester is about how to kill microbes in different ways.
Favor like a, so they favor like a,
Say that again.
on helmets, do they favor like a certain intestine? That's all.
They, yeah, so they would, let's see what's a better, probably this is a better picture. All types of parasites, if they have a favorite part of the intestine, it's small intestine, because that's before we've absorbed nutrients. So it's like you're hanging out there and there's dissolved nutrients all around you. And it's great, it's warm and it's watery. By the time they get in the large intestine, we've absorbed the nutrients. Hopefully, we've absorbed the water. And there's just feces left. So most parasites will go into their survival form in the large intestine, but they're more active or healthier form in the small intestine. So for worms, they would be like mating in the small intestine. And then by the time of large intestine, there would be eggs, but not crawling around worms. So the way you diagnose is you look in the feces and you look for the eggs, or you look for the cysts under a microscope.
Do they actually like reproduce, or do they actually have like a?
It depends on the specific worm type. So the tapeworms, I am less sure if they are mating in there, but the roundworms are. Flukes, I'm not sure. Good question. Did you guys learn a lot about worms in Bio 2? It depends, like, were you in the same class as Bio 2? Do you remember? It depends on the year and it depends on who teaches it.